Hi. Last time we finished talking about Royal Society, followed by an extensive discussion on Nobel Prizes in physics, primarily concentrating on the uh, Standard Model as well as the Big Bang theory and some assorted findings in physics. So today we'll briefly talk about chemistry and delve introduced to talking about physiology and medicine Nobel Prizes. And if time permits, we'll uh, get into the new kid on the block, the breakthrough prizes. Okay, now it's always fun. Chemistry is basically elements, so we'll start out with the old favorite that I'm sure most of you have heard about.
There's antimony, arsenic, aluminum, selenium, and hydrogen and oxygen and nitrogen and rhenium, and nickel, neodymium, neptunium, germanium, and iron, americium, ruthenium, uranium, europium, zirconium, lutetium, vanadium, and lanthanum, and osmium, and astatine, and radium, and gold, protactinium, and indium, and gallium, and iodine, and thorium, and thulium, and thallium. There's yttrium, ytterbium, actinium, rubidium, and boron, gadolinium, niobium, iridium, and strontium, and silicon, and silver, and samarium, and bismuth, bromine, lithium, beryllium, and barium. There's holmium and helium and hafnium and erbium and phosphorus and francium and fluorine and terbium and manganese and mercury, molybdenum, magnesium, dysprosium and scandium and cerium and cesium and lead, praseodymium and platinum, plutonium, palladium, promethium, potassium, polonium and tantalum, technetium, titanium, tellurium and cadmium and calcium and chromium and curium. There's sulfur, californium, and fermium, berkelium, and also mendelevium, einsteinium, nobelium, and argon, krypton, neon, radon, xenon, zinc, and rhodium, and chlorine, carbon, cobalt, copper, tungsten, tin, and sodium. These are the only ones of which the news has come to Harvard. And there may be many others, but they haven't been discovered.
Okay, so there is a more recent version by Daniel Radcliffe from the Harry Potter fame, but his is not as musical, so I thought we'll go back to the original. Okay, so after this, we'll get started into some real science. And now, obviously, all of us are old enough to remember this, the classic scene from The Graduates. But there is a long history behind it, which is equally the start of chemistry. And this is, I'm sure a lot of you have heard the word Becquerelite, but this was something called the Beckelizer. So this was actually a headline in New York Times, So this is chemistry formula that reads like, basically it reads C7, H38, and O43. Okay, and this was, so this was a formula for Becquerelite that they decided to publish for it's going to be a dramatic change. This was synthesized by a Belgian chemist, okay, and the signs that are included are some of the mathematical symbols which were coined by Bertrand Russell. The inverted U applies. Okay, V stands for or. So either or. And so these are the formulas that Bertrand Russell had proposed in the Principia Mathematica type thing. So, and these are the inventions that came out of Becquerelite. This was done by a Belgian photosynthesist. He was an inventor par excellence. He had already sold some research to Eastman Kodak Company, and later on worked on formulating, from from hydrocarbons, this formula, and that's the next is history. So a lot of the material that that he coined with, had varieties of practical uses, and that was the start of plastics as we came to know it. Obviously, this was not Nobel Prize winning discovery in the minds of the chemist in Sweden. the history of obviously the Marie Curie was a repeat winner. Okay, and she synthesized tons of pitchblende, a highly complex mineral which is made of 30 different elements of unknown substances, and they succeeded in isolating a very small quantity of radium which had spontaneous radioactivity, and that was a quite dangerous work. They were dissolved in acid, leaving the other elements in a sludge underneath the pot. Okay, and this was they used the radiation measurement to mount the tiny amount, and in July of 1898 they published the results of a new element which chemically it acted almost exactly like bismuth, but the big difference was it was active, and they named it polonium. A second publication of a new element that they discovered, which was even more radioactive, which they discovered radium from a Latin word for ray, and that started the so-called radium craze, and everything was radium, including radium water. Now, obviously, the watch dials, which were, caused the women who worked in those plants, they got cancer, and then even, I think I had some slide which I think for uh, length of time I did not even radium condoms were marketed, radium toothpaste, and so that was the start of the radium craze before people realized that it was toxic and highly dangerous. And so that was, the start of the radium discovery. And then obviously her daughter who helped in the X-ray during the First World War, and later on they went on to get their own Nobel Prize. So just because this is out of sequence, but I just thought I'd place it in because to get the family history out of the way. And this was, and they managed to work on radioactivity of converting aluminum to phosphorus. So this was basically alchemy in the tank. They also worked on chain reaction, but they thought protons were released. I talked a little bit about it when we talked about Chadwick and discovery of neutron. Okay, now some of the other people have suggested that this chain reaction thing should be kept secret, but it was published anyway for everybody, and that led to the rise of atomic age, and we talked about it in the earlier class. There's some of the more notorious, but at the same time useful discoveries were what came to be known as Haber-Bosch process. This was, I'm sorry for the dark slide, but it was a Nobel Prize in chemistry, which was controversial to say that given to Fritz Haber, and followed by another prize given to an engineer who worked in collaboration with him named Bosch, and he got the Nobel Prize in 1935. So this was a long sort of object because the fertilizers came from natural product of guana from Peru, and this was a highly sought out product. The bird poop that was used as fertilizer, and the idea that was dreamed for and sought after by chemists of fixing nitrogen from the air. But there was a hard element to fix. It was relatively inert, did not react. So using high pressure and catalyst, as it is illustrated in this thing, as you can see, that where is catalyst bed, and that is heated with compressor and cooled off. So that was the founding of the Haber-Bosch process. And this was one of the most technical advances of the 20th century. And this enabled the mass production of fertilizers and managed to feed huge number of people and avoid starvation all over the world. So that, and obviously a well-deserved Nobel Prize that was given for the, but Haber has a dark history that even though he was the Jewish ancestry, he was a strong patriotic German, and then during the war, he decided to help the fatherland. And you can see his quote, during the peacetime, a scientist belongs to the world, but during wartime. So the idea of nation state was absorbed and was in his DNA, to use the cliche. And he was the founder of lethal gas like phosgene, chlorine, mustard gas. So the chemical warfare became the first major disaster of the global war during 20th century. And there were about 90,000 fatalities from, and 1.3 million casualties of gas attack. And this was, a dramatic departure to the warfare and the widespread use of chemical warfare, and gave rise to the chemist war as the World War First. So one can say that was before the nuclear weapon, the poison gas was used by all the belligerent countries and violated 1988 Hague Declaration. Okay, and again, unfortunately, that was not duplicated in the Second World War, but obviously the stockpiles were there to be used just in case, and I'm sure they're still secretly stored by both major powers. And unfortunately, with the rise of Nazis, Fritz Haber came to a bitter end, and he had to run away, and I think he died before the Nazis assumed complete power. But that's we're digressing. The second interesting prize, which came to backfire on humanity, was I don't think a Nobel Prize, but the discovery is worth mentioning. Obviously, a guy named Thomas Midgley, and he was responsible for two big environmental catastrophe. The rise of automobile age. He was founder in adding lead to the petroleum to prevent the problem of knocking, which was a big disaster for the major car manufacturers. And he was able to solve the so-called knocking problems. The engine damage was occurring. So by changing the octane number of the gas, and was able to, so varieties of elements were tried. Initially, selenium was used, but that was rather expensive. So he started using lead, and this was, now there were some physicians who were aware of the potential danger. And the founder of the occupational medicine, a lady doctor named Alice Hamilton, had already alerted this, but the Ethel Corporation was economically very powerful, and the lead use had continued for almost 40 or 50 years. Okay, and so starting from 1923, this was the usage continued. Midgley was a strong proponent of it, and then there was, in spite of the workers being killed due to exposure to lead, tetraethyl lead, at Standard Oil Refinery in New Jersey, but still continued in general sale, and the lone voice of Dr. Alice Hamilton was ignored, and Midgley was actually suffered lead poisoning himself, and not to be outdone, he was the discoverer of carbon tetrachloride, and which had a lot to do with the depletion of ozone layers. Okay, so these were the two discovery of fluorocarbons, as you can see from the formula here. Okay, and the lead. So this was, and it took a long time for humanity to be aware of the danger of the ozone depletion, and then a scientist named Crutzen coined the word Anthropocene age, which was, again, and the Antarctic ozone depletion that was pioneered by chemist Thomas Midgley's discovery. Okay, so later on, the finding that was discovered by the chemistry was for the work in atmospheric chemistry and concerning the formation and decomposition of ozone. So it took several decades for science to come up with the cause. Okay. Interesting, digressing to the basic chemistry. We already talked about Niels Bohr, who was the founder of the quantum theory earlier. And we talked a little bit about the electronic shell theory. And he had used, when he was presenting his, some of his associates had now the periodic table that Mendeleev had proposed had some gaps. And while he was presenting this lecture, that the element that was theoretically proposed, was discovered, and for the atomic number 72. And appropriately, with Bohr's national origin, this was named hafnium in honor of Denmark, hafnia meaning Copenhagen. So he was able to tell the story that what he had theoretically predicted based on the electron shell properties, this was discovered shortly after. So it was a very dramatic finding that he was able to share with the world at large. Okay, now no talk on Nobel Prize is complete without the founder of physical chemistry, i.e. Linus Pauling. He was a giant in his own thing, and he was originally from Oregon and did most of his work at Caltech, or California Institute of Technology. And he was, the so-called founder of physical chemistry, and was tremendously productive. And he had published more than 1,000 articles, and in this classic work on physical chemistry. The book was translated in 13 languages, and he was absolutely a prolific researcher. And the book that he had written was The Nature of Chemical Bond, and as to how the electrons are shared, in the various orbits, and the combining sort of physics and chemistry. The two disciplines were put on a more solid findings. So his work was not just in physical chemistry, structural chemistry, analytical, organic and inorganic chemistry, and as well as in biochemistry. So theoretical physics, primarily quantum theory and quantum mechanics, were combined with the atomic and molecular structure and chemical bonding. So, and he'd also had his forays in metallurgy and mineralogy of atomic structures of various metals and minerals. Okay, so a large number of inorganic compounds, that he had published the chemical formula. And then later on, he had branched out into what later on came to be known as molecular biology, including structure of hemoglobin, the formation of hemoglobin formula. So that was, and then later on worked with epidemiology, biomedicine. So and also was able to show the abnormalities of sickle cell. So you can just say that his impact was larger than life. And now it just goes to show that even Nobel Prize winners have their own limitations sometimes. And he had, in a hurry, there obviously at that time the DNA structure was to be elicited, and everybody thought that that this was protein, and he had rushingly worked on the DNA formula, but had made some basic physical chemistry mistake in terms of putting the ACGT on the outside rather than the sugar phosphate bond. And his paper was taken to Cambridge by his son, and Watson and Crick spotted this chemistry mistake and were able to correct that mistake and rush their discovery. So that's an interesting side story on history of chemistry. Now, the hormones were eminently popular, and the chemical research on hormones dates back to long time. As to, you can just see that the litany of hormonal discovery goes back to 1947, and it has been recognized and awarded a number of times, including, let's start from the bottom, the pituitary hormones and carbohydrate metabolism, as well as the metabolism of sugar molecules and glycogen, adrenaline, and oxytocin, the so-called love hormone, insulin, and all of these have been recognized, that it took some hard work with chemistry and including neuroscience, the basic chemical, the neurotransmitter, acetylcholine, and that they've all been recognized by the Nobel laureates. And we'll talk about some of this individually when it relates to health, especially structures such as cholesterol metabolism and some of the neurotransmitters are, and obviously the IVF. So there's a long list of involvement in chemistry and physiology that has been recognized as Nobel-worthy discovery. Okay, moving on to the more interesting, this thing where medicine and biology, and the one of in addition to Marie Curie, the one person who received 2 Nobel Prizes is mild manner chemist par excellence, Sir crystallographer, is Sir Frederick Sanger, who in as early as 1958 was based on the work by the Bragg father and son as thing, because X-ray crystallography was extremely difficult and required very minute determination of the atomic structure of, first of all, they had to crystallize a protein and mount it on a small object and look under microscopes with X-rays and to determine the atomic structures. And initially, he was able to determine the amino acid sequence of insulin, which is a very large molecule, with a huge atomic, with a very complex atomic structure with different chains. And it was very laborious and detailed work. And for that, he was recognized. The second work that he was recognized was several years later for a DNA sequencing method. Okay, and the sequencing method became the workhorse of electrophoresis, and this was used by Crick and Watson, and later on for deciphering what came to be known as amino acid sequence and DNA code. You can just see as to how, and this was, like how by adding, various nucleotides and tagging them with radioactivity, okay, and how they migrate on X-ray film with its so-called, because the atomic weights of these nucleotides are different, so they migrate at a different rate on gel electrophoresis, and the laser detection of this thing can be mapped. So this was the came to be known as a Sanger method of nuclear sequencing. So this was obviously changed the molecular chemistry dramatically. Okay, moving on to carbon decay, because we talked about earlier that the age of Earth, when we talked about radio in Royal Society, there was always a controversial starting from few thousand years to billions of years, and that went back to even in the 19th century, the observations that were done from geology and evolution were in contrast to what the chemists and the physicists were stating, and that required what is known as the various isotopes of carbon, which is ubiquitous in a plant and human life, and the isotopes decay at different rates. So a chemist named Walter Libby, worked. It took as late as 1960 to use carbon-14 decay in determining the age of various elements. As you can see that how the decay curve is different, and based on radioactive half-life, you can just see that half the elements is decayed. So based on that, and plants when they consume due to sunlight, and based on the isotope determination, one can determine, the age of the elements, and this was used for. Okay, let's see if we can play this.
It is until a singular discovery in chemistry changed the course of history. Welcome to The Day Tomorrow Began, the genesis of world-changing ideas from the University of Chicago. In the 1940s, the discovery of a new isotope, carbon-14, was of particular interest to University of Chicago chemist Willard Libby. Libby was the first to suspect that all living organisms would absorb carbon-14 from the atmosphere. After the organism dies, the carbon-14 slowly starts to disappear, like a countdown clock. Libby theorized that by measuring the carbon-14 left over, he could determine the approximate time of death. To test this, Libby measured samples of known antiquity: an Egyptian funeral ship, a Dead Sea Scroll, and a charred piece of bread from the ashes of Pompeii. It worked. The radiocarbon revolution had begun. Carbon-14 dating transformed fields from archaeology to oceanography. It proved civilization originated simultaneously around the world and determined when the ice ages began and ended. Today, carbon-14 dating is being used at the University of Chicago and around the world to power new scientific discoveries, from revealing how our bodies work to understanding changes in Earth's climate. As Nobel Prize nominator Kenneth Pitzer wrote, "Seldom has a single discovery in chemistry had such an impact on the thinking in so many fields of human endeavor." To learn more about this and other
Okay, so they worked at University of Chicago, about the decay, and the carbon-14 has a half-life of about 5,700 years. So that means half the samples will change into other atoms. So that's the concept of decay. So this has been useful in determining the age of the Mayan calendar, to the Iceman Otzi, and the so that it's been a dramatic improvement in, and now obviously the technology has been improved with particle accelerators, in the 70s, and so this technique has become, but it has its own limitations because that carbon-14 decay is relatively limited. So other isotopes are used for longer determination.
world-changing.
Okay, and now the staple of this is, I've picked out just some of the more interesting things. So the staple of forensic medicine, which PCR, which everybody who has watched the NCIS show or detective shows is aware of PCR. And this was discovered by a chemist working for CETA's lab. And in this was what he did was try to exponentially expand the DNA amount that, so we can start out with this tiny amount and keep adding, by heating and cooling. So initially, let me just go back to this thing, that the samples are heated up to 95 degrees. So DNA is denatured and separated. The two strands are separated, and then it is cooled off to 55 degrees. And at the same time, DNA primers, which are the fragments of A, C, G, and T, are added and multiplied. So it keeps duplicating it. And as long as you keep repeating the process, the amount of DNA that is available multiplies from 2 to 4 to 16 to 32. So after running several cycles of heating and cooling, or like denaturing and annealing, that's the technical term for it, you get a large amount of samples. Okay, and that became the workhorse about for diagnostic, from crime scene, from medicine, and for identifying the smallest amount of samples. So this has been for collecting, analyzing fossils, and with the tiniest amount. So with this, it is possible to, get the idea as to the amount that is present. and also it's used for dramatical, for medicine, and protein design. So it has a wide impact. And Kary Mullis was recognized for his discovery, and there were some controversies about the patent of this technology, and that was his one and only contribution. And finally, he kind of disappeared from the scene and went somewhat off tangent, supporting various poppycock type of theories. Another woman who has been the giant in crystallography was an English lady named Dorothy Hodgkins, and she was mentored by various other chemists at Cavendish Laboratory, and she was crystallographer par excellence and determined the structure again of penicillin, vitamin B12, insulin, and so she was she was a product of the empire and was mentored by various other crystallographers, named Burnell, and she taught the Prime Minister Maggie Thatcher, who was before she changed her name, was Maggie Roberts, and she was a socialist and somewhat of a sympathizer for the Communist Party, and was denied entry to the United States because of her previous political leanings, and eventually she was one. And I mentioned earlier when we talked about the Copley Prize medals, that she was recognized for one of the few women to receive Copley Medal for Science. Okay, lesser known discovery is 1978 was a self-funded scientist, chemist par excellence, who was assisted by his able assistant, who was not recognized for her role, named Peter Mitchell. He had an independently self-funded laboratory, and he worked on the chemical mechanism of photosynthesis and came up with what is known as a chemiosmotic theory, which is less well known. So this implies the chemical mechanism of photosynthesis and mitochondria as to how ATP is utilized. We can just say this is something called the proton gradient, which goes through the nicotinamide acid dehydrogenase, picks up the hydrogen molecule and goes through various complex changes through coenzyme and complex 1 to 3 to 6, and the hydrogen or the proton is transferred across the membrane in the case of plants and in mitochondria. So the ion exchange takes place. So this is the electrochemistry that he proposed, and later on the theories was considered, what the ideas were considered controversial as to how the excitement takes place, and this is the excited molecule and enters the chlorophyll, and then the reaction takes place. So it's a fairly complex molecular transfer that he proposed, and later on has been proven to be correct, but it's less well known than it needs to be. So, and this, and even there may be an element of quantum transfer that has been proposed by various other people who are extending his work. And so there may be more to it than we realize that there of the chemical role. Another interesting discovery that needs to be mentioned is something called a gene fluorescent protein that was recognized for Nobel Prize for discovery. And we'll see if I can.
Roger developed variants at the conference. There's a type of jellyfish known as the crystal jelly. A researcher named Osamu Shimomura began studying it in the 1960s, and he discovered a protein in the jellyfish that glows bright green under a UV light. It came to be known as the green fluorescent protein, or GFP for short. Years later, a researcher named Martin Chalfie heard about the green fluorescent protein at a conference.
I got very excited and immediately ignored the rest of the seminar and just sat there fantasizing about what would happen if we could possibly take this protein and allow it to be made inside of our worms and look at how things were being made.
At the time, he was studying these small organisms called roundworms. There's all this activity going on inside of an organism that's completely invisible to researchers, even with a microscope. Martin realized that he could use GFP as a way to map the inner workings of a roundworm. This was a big deal. Like the invention of the microscope, the X-ray, or the MRI, green fluorescent protein opened up a new world for scientists. They could suddenly see what was happening inside of a cell that had been completely invisible before. But the green fluorescent protein wasn't perfect. It would lose its glow over time, and it wasn't the ideal color for all studies. Here's where UC San Diego researcher Roger Chen enters the story. Roger developed variants of GFP that were brighter and came in a variety of colors beyond green.
And we then had to come up with names for these, and upon examination of the Crayola website and other consultations, we realized that the simplest, instead of giving them numerical names, were names like monomeric banana, orange, tandem dimer tomato, tangerine, etc., all the way out to plum.
This beach scene in a petri dish was made using some of these colorful variants in Roger's lab at UC San Diego. But these glowing proteins aren't just pretty. They allow researchers to mark and observe more than one thing at a time. Researchers at Harvard University used them to color nerve cells in a mouse's brain to see how they form a network. The experiment was called the Brainbow. Today, scientists at some of the world's leading hospitals use green fluorescent proteins to study all sorts of diseases. UC Davis uses it for HIV research, while UC San Diego used GFPs to track breast cancer tumors, and Mass General has used the technology to better understand Alzheimer's disease. When Osamu Shimomura began his research, he didn't know that this jellyfish protein would lead to a scientific revolution. He was just trying to answer a basic question about how our world works. And in 2008, these three researchers won the Nobel.
Okay, so you get the idea type of thing. So and so this has been a dramatic breakthrough, and it's can be used for fun experiments, and people can color their own goldfish in a bowl to glow at nighttime.
Prize for.
Okay, now another interesting prize that deserves a nod, which is ribosomes are the structure which transfer. Are part of the they're in the cytoplasm in plentiful, and the ribosomes are they're the transfer RNA and messenger RNA. These are the big parts of the workhorse of the molecular work, and the three people were working on the molecular structure of atomic structure of these ribosomes, which form the proteins. And there are different types in the so-called eukaryotes and prokaryotes. There are different types of ribosomes that are present. So the race was on between the Israeli scientist Ada Yonath and Venki Ramakrishnan was working in Cambridge. So to determine the ribosomal unit comes with a different atomic subunits that come up with, and you can see that that comes in, the prokaryote, which are the earlier bacteria without the nucleus. They have like 5S, and S is called Swedberg. That's the way of measuring the atomic unit, and 23 RNA and 16. While in eukaryotes, that's a larger molecule, and which is 5S and 23 and 18S. So that's the different weights. You can just see 50 and 30, 60 and 40. So they are slightly different structures. So they had to work on crystallography, and so this was their work that as to how the messenger RNA transferred through the ribosome units, and is like a cache register plate as the amino acid sequences come, and it is coded and put together in the various ribosome. This is like the factory inside which the ticker tape goes back and forth and creates. So this was the big discovery that led to the Nobel Prize for determining chemical structure. Now, just as a side note, Venki Ramakrishnan went to the same university that I did in India, and later on got a PhD in physics, and could not find a postdoc position in US, and later on switched fields from physics to biology, and worked at Cavendish Lab in Cambridge, and the rest is history. And he was one of the few people who was the head of the president of the Royal Society. And I mentioned that earlier, I think. Okay. Now, another interesting prize, which we'll jump directly, because some of the other chemistry Nobel Prizes are extremely complex and about various chemical reactions, and they may not be of interest to most of us, type of thing. And now the discovery of the battery, which was the lead acid battery, has been around since the days of Sir Humphrey Davy to 1958, and nickel cadmium batteries were around in the early part of 20th century. And with the oil embargo, it became the need for generating more power. So John Goodenough developed a lithium battery with a cathode. It's the negative of cobalt. At the molecular level, lithium ions can go back and forth. This gives the higher voltage than the previous batteries. And so this was led to the lithium ion battery, which became the workhorse of various electronic devices, and it is used for cell phones. Okay, and he's got to be one of the oldest Nobel Prize winners at age 97. He was born in 1922 and died in 2023. So even as 97, that's when he received his Nobel Prize for his discovery several decades ago. And this is the principle of the lithium-ion batteries, the earlier version. Okay, and that later on, I think titanium was replaced with cobalt, and the Japanese made it this practice more usable by improving the lithium battery, and the Sony Corporation. And sometimes the danger was some of the recalls were done due to potential for catching fire, and it still sometimes occurs even in Tesla automobiles. So that is that's one of the dangers that is still we're dealing with. So the lithium pulls the electrons from the graphite solution, which is there in between, as a barrier type of thing, and a positive charge lithium ions can form, and and they go back and forth, and it distributes. So that's requires a various amount of the physical chemistry that takes place between charging and recharging. Okay, the more important Nobel Prize, which we have talked about, is this. Let's just play this.
The CRISPR-Cas9 system is a tool for cutting DNA at a specifically targeted location. The technique has already revolutionized gene editing, but scientists are always looking for new possibilities. So what else can CRISPR do? Since being discovered in a bacterial immune system, CRISPR-Cas9 has been adapted into a powerful tool for genomic research. There are two components to the system: a DNA cutting protein called Cas9 and an RNA molecule known as the guide RNA. Bound together, they form a complex that can identify and cut specific sections of DNA. First, Cas9 has to locate and bind to a common sequence in the genome called a PAM. Once the PAM is bound, the guide RNA unwinds part of the double helix. The RNA strand is designed to match and bind a particular sequence in the DNA. Once it's found the correct sequence, Cas9 can cut the DNA. Its two nuclease domains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, the fixing process is error-prone and often inadvertently introduces mutations that disable the gene. This makes CRISPR a great tool for knocking out specific genes. But making double-strand breaks isn't all CRISPR can do. Some researchers are deactivating one or both of Cas9's cutting domains and fusing new enzymes onto the protein. Cas9 can then be used to transport those enzymes to a specific DNA sequence. In one example, Cas9 is fused to an enzyme, a deaminase, which mutates specific DNA bases.
Okay, so this gets to be a little bit more technical than we want to be, but this was the technology that it took several years for it to be worked from bacteria to eukaryotes, means the cells with nucleus, and was a big controversy between MIT and West Coast and for patent rights, and is well summarized in the biography of Jennifer Doudna by David McCullough. And I'm sure some of you have read the book. Okay, and moving on to the various elements, that are named after scientists, which is, I just thought it would be of interest, that several elements have been named for scientists, which includes for Niels Bohr, Marie Curie, Einstein, Fermi, Lawrence, all the people that we talked about, Röntgen, Rutherford. So these are all the elements that are named after. Okay, and even then, and the other ones are, I'm sorry, let me go back to, which includes Dmitry Mendeleev, Alfred Nobel has been given, and then this one, I don't know who that is, Yuri Oganessian, and John Gadolin, I know it's gadolinium, and some other person, which I have no idea who that is, Georgi Flivero. So the varieties of elements have been named after scientists from the periodic table. Okay, is the periodic table complete? We don't know. Okay, a chemist, physical chemist, par excellence, was a person named Glenn Seaborg, who is at the bottom, and he was kind of a giant he worked with Lawrence. He was from the Midwest and had a PhD in chemistry and worked on the atomic project and was instrumental in isolating plutonium in 1941, which became the second bomb. Okay, and in addition to plutonium, they discovered various other elements which are beyond uranium, which are primarily synthesized in the laboratory and short-acting: americium, curium, berkelium, californium, einsteinium, fermium, mendeleevium, nobelium, seaborgium. So this is, so he was a giant and he was later on chairman of the Atomic Energy Commission, and was successful in truly achieving the alchemist's dream of converting bismuth to gold, and the so-called classical philosophical stone. But it was horrendously expensive procedure to do, so it never really, I don't think the idea of creating gold in laboratory are still a dream that for scientists. So this is, the varieties of elements were discovered by Nobel Prize winners, but Glenn Seaborg was, was a giant in the field for, and another person was his associate is called is a guy named Ed McMillan, and he had discovered varieties of neptunium, plutonium, such that the actinide and lanthanide series. And we already talked about Emilio Sagre earlier and the Curies, so that's a rehash. So there's a huge history. And another prize that is of interest is something called the directed evolution, because evolution is a slow, taking thousands or if not millions of years. But a chemist named Francis Arnold in Caltech had done something called enzymatic directed evolution, which is has been a dramatic change in some of engineering and creating, literally mimicking natural selection. So let's play this.
Francis Arnold, George Smith, and Gregory Winter have won this year's Nobel Prize in Chemistry. And this year, we've got ourselves a Nobel twofer. The award is going to two distinct but related fields. Let's start with the first half of the prize. With this year's Nobel nod, Francis Arnold became the 5th woman to win a Nobel Prize in Chemistry. She's recognized for her invention and development of a technique called the directed evolution of enzymes. Let me explain. Enzymes help enable the chemical processes of life, such as metabolism. We also rely on enzymes to help us manufacture medicines and detergents. Using regular old evolution, nature has made enzymes that work, but there's always room for improvement. Evolution is not necessarily optimization. Realizing this, chemists have tried to design better enzymes that are more active than their natural counterparts, or enzymes that could thrive outside their normal biological environment, say in a commercial reactor. Rather than assume chemists could design the best enzymes on their own, Arnold developed a way to enlist nature's help. In Arnold's directed evolution, she first engineers mutations into the genes that code enzymes. She then introduces a variety of these genes into a bunch of bacteria. Using the modified genes, the bacteria make modified enzymes. Chemists then test for which of these modified enzymes are best suited for the job we want them to do. Arnold gave a seminal demonstration of this in the 90s by modifying the enzyme septolysin. Like most enzymes, it works in water, but Arnold directly evolved a version that works in an organic solvent used in industrial reactions. As Klaus Gustafsson of the Nobel Committee put it, it's like Darwinism in a test tube. Today, directed evolution has produced enzymes that are thousands of times more active than enzymes found in nature. And it has yielded enzymes that help us create medicines and biofuels. On to part 2 of Chem Nobel 2018, which goes to George Smith and Gregory Winter for their work with phages. Phages are viruses that essentially hijack bacteria. Phages unload their genetic payload into bacterial cells, and those genes trick the bacteria into making more phages. Total virus move. In the mid-80s, George Smith harnessed phages to sort of mass-produce and screen peptides and proteins of interest. First step, stick a gene of interest into a phage. That phage builds the corresponding peptide or protein and plants it on its surface. Smith then used an antibody lure to pluck that particular phage out of a pool of other phages. And once he's caught the phage, he's now got the protein and the genetic information that helps create it. So Smith introduced this new tool called phage display to explore biochemistry. George Winter's work brings this year's prize full circle. Building on Smith's phage display, Winter got phages to plant human antibodies on their surfaces. Using directed evolution, he then created multitudes of mutated antibodies. And he looked for the ones that would bind best to target proteins, proteins that are, say, involved in human diseases. Here's how American Chemical Society President Peter Dorhout used the science behind this year's prize. ACS also publishes CNEN, which is my employer.
The work that these three laureates have been involved with over the years has really helped advance our understanding of how fundamental chemistry of complex systems can be learned and ultimately controlled.
And as we hinted at earlier, learning this fundamental chemistry has already played out in real-world applications. Winter's work led to a drug based on a human antibody that can halt inflammation in the body, and that drug was approved in 2002 to fight rheumatoid arthritis. You may know it as Humira. More recently, Arnold co-founded the company Provivi, which uses directed evolution to develop enzymes that protect plants from insects. Incidentally, that company was also one of CNN's 10 startups to watch in 2015.
Okay, so you get the idea about, so that's, for the sake of move
So-
right along. Okay, and then we're moving on to, we'll jump on to this year's Nobel Prize. So before we pause.
Have you ever heard of a quantum dot? The discovery and development of quantum dots was awarded the 2023 Nobel Prize in Chemistry. Today, quantum dots are found in television screens and LED lamps. Their lights can also drive chemical reactions and can even illuminate tumor tissue for a surgeon. Quantum dots are particles so tiny that their size is measured on the nanoscale. To give you an idea of how small that is, a quantum dot is about 1,000,000 times smaller than the head of a pin. Strange things happen when particles are this small. For example, quantum dots emit different colored lights, depending on their size. Larger quantum dots emit redder lights, while smaller quantum dots emit bluer light. This discovery opened up the field of nanotechnology, where the properties of a material change based on its size. These characteristics have given us new possibilities to discover materials with extraordinary properties.
Okay, so this is, so basically the start of nanotechnology and material science. So, that can be illuminated by UV light, and so that can be excited to different energy level and semiconducting properties. And so it's a dramatic change in terms of how material science can progress. So I think we are done with the things. I will pause at this stage. Okay, we'll jump into physiology and medicine. Now, I'm trying to just give a big, major outline because this is of interest to all of us with health and science. So the biggest discoveries, that one can say that medicine has gone through, huge advances, starting with, we had to eliminate some of the big advances that took place was treatment of diabetes, hormones, antibiotics, and preventive care, surgery advances, treatment of mental disease, and artificial joint replacement and bypass, and varieties of transplants, in vitro fertilization, curing cancer, treatment, especially for hematological cancer, and the genomic revolution. So these are some of the major discoveries of the 20th century, which has increased our lifespan. And in terms of testing, we have come up with computer scanning, not just X-rays, to varieties of genetic code, treatment of heart disease, and genomics, along with the advances in general anesthesia. Obviously, so now this goes way back before even the Royal Society that we talked about in the day one. As to I would lose my cardiology credentials if I didn't talk about cardiology. And William Harvey was born even before the Royal Society. In fact, Alphonse died before the Royal Society came into existence. And he discovered, after traveling to Padua, and with careful observations, threw away the whole Galen's concept of circulation and established as heart as the center. Okay, now he didn't have all the details worked out because obviously microscopy wasn't around and capillaries were not known. So his model had some deficits, but he was considered the father of cardiology, so to speak, and he was a physician of repute in Britain of that era. And in addition, he had also ventured into embryology. And obviously, in Latin, his famous quote is ex ovo omnia, means life originates from the egg. So there was nothing like a spontaneous generation that took place or homunculus. So you can just say that this was the start of more a scientific basis of medicine. Now, interestingly, the first Nobel Prize in medicine, which had a huge impact, was given for physics, which was X-rays. And X-rays, obviously was an accidental discovery by William Röntgen that we talked about, at the start of our physics talk. But was very soon people became aware of the medical use. And an enterprising medical student named Emil Kirby, started using it for treatment of cancer. And because obviously it takes a while for the Nobel Prize to get awarded. And even in 19th century, he started using X-ray tube to bombard cancer. So this was the start of medical uses of X-ray radiation. Okay, and at the same time, as the invention became very worldwide use very quickly, and people started using it for just about everything, including trying to find correct shoe size, and was marketed became like a new craze before realizing the dangers, and it took several years for it to become regulated. Okay, and even though the drugs regulation came into existence in 1908 after some disasters that took place, with over-the-counter drugs, and but the devices regulation still became like a Wild West. Okay, the varieties and the Nobel categories go in various fashions. Initially, the infections were the biggest bane on humanity, and insecticides, such disease such as malaria, syphilis, were tackled first. Later on, the body's resistance, immunology came into favor. Later on, cancer, and then genetics and various medical devices. So varieties of categories have been in and out of fashion. So, and if you divide them further, infections, cancer, genetics has become more popular, and neuroscience is always up, right up there on the list. Okay, and vitamins and hormones. So varieties of categories which are relevant. Okay, the first Nobel Prize was given for treatment of diphtheria. Diphtheria is a rubber-type membrane that takes place due to infection in children and can be very fatal disease. And initially, the vaccine was discovered based on serum by collaboration with a Japanese scientist and a German scientist named Bering. Okay, and for whatever reasons, the Germans was recognized for its discovery, and he had an able assistant who will figure larger a few years later named Paul Ehrlich. Okay, and then obviously, that right at that time, the commercialization of vaccine came into vogue, and but that vaccine has been the cornerstone of, and almost eliminated the potentially very serious disease, and now is combined in all childhood as a triple vaccination of diphtheria, tetanus, and and pertussis, which is commonly known as whooping cough. So 3 shots are given. This is the standard recommendation. Later on, we added with measles and as well. So the four several vaccines that you get during the first 3 years of life, and that has eliminated a lot of these diseases. The deadliest organism is not a virus or bacteria, but the smallest insect, mosquito, and obviously So this was the Nobel Prize for proving malaria belt, which is in the tropics and kills millions of people still, and this was given a Nobel Prize to a British physician and scientist named Ronald Ross, who worked in India, in the colonies, and was able to isolate the malaria parasite inside a mosquito with very meticulous detection. Now, his discovery was primarily in birds and was able to reproduce the disease in birds. At the same time, in Italy, a physician zoologist named Giovanni Battista Grassi was working with his colleagues. He established the malaria parasite cycle in humans. But for whatever reasons, his work was ignored because Rossi was politically savvy and had accused Grassi of fraud, and the controversy or the argument was sent to the founder, to Robert Koch, and who was appointed the arbiter, and he leaned towards Ross, and so Ronald Ross was the unique Nobel Prize winners, and so these are some of his quotes, I think so. But he was not the easiest person to work with. So discovery involved several people, and even when he was recognized, he did not acknowledge the contribution of his mentors, a giant in tropical medicine named Sir Manson, and then later on, some of the parasitology was a big field of research, hot field of research at that time. Okay, and now at the same time, tuberculosis was equally prominent, and unlike cholera, tuberculosis death was considered very romantic and dramatic, and a subject of various novels. And the rich people went to sanatoria in Switzerland, and it was considered the pale suffering death, which has been memorized in various books, okay, by Thomas Mann and Virginia Woolf and other laureates, okay. And tuberculosis, all of you know that it affects the lungs, but it can also affect various other organs, including skin, okay. And it's called lupus vulgaris, so you can just see that the face is deformed, and unlike the lupus, which is autoimmune disease. And this was, and people borrowed the idea from X-rays of using radiation and ultraviolet light for treatment, and lo and behold, they were able to cure it. And so light therapy has been in vogue and in various other medical system, including the Indian Ayurvedic system, and light has been beneficial. And this was marketed by various other known scientists, as well as the con artist, the one that was lesser known is an Indian Ayurvedic specialist who migrated to the United States and had marketed this as a spectrochromograph. And the AMA went after him with their full political power. And but obviously, Dr. Finson used this effectively to treat skin tuberculosis. And tuberculosis can also affect joints, spine, and hips before the milk became pasteurized. And now Dr. Robert Koch is well known as the pioneer of microbiology and discovered various organisms, including anthrax, cholera, vaccine, tuberculosis, and was the founder of bacteriology with the well-known Koch's postulates, okay, which we'll jump to right away before we come back to it. Koch's postulates are basic procedures used in epidemiology and medical research as to identify the organism, okay, obtain culture it, grow it, but inject it inside experimental animals, and harvest the organism. So these are the various scientific thought that Koch got postulated, and that's the foundation of bacteriology as we know it. And he was correct, and he was a giant in his field, and traveled all to Egypt and to India to discover cholera, and was a big force who was in direct competition with Louis Pasteur. And he had discovered this testing for tuberculosis or tuberculin serum, and it said that this may be useful in abating the disease, but people were looking for hopes and decided that it was blown out of proportion that tuberculosis curing was a cure, and people started en masse to receive tuberculin therapy, and that did not pan out to be. And Arthur Conan Doyle was suspicious about this so-called tuberculin remedy, and he investigated it, and he sort of went after it with scientific vigor, and so that this actually did not, fail to pass. Wrote a book about him saying that this was not what it was all cracked up to be. And in Koch's defense, he never claimed that it was a cure, but the publicity ran out of him. And again, his personal life was was interesting. That after a long 25 years of marriage, he married a 17-year-old, and his able assistant was a guy who named Petri, and that was become the cornerstone of bacteriology, named Petri dishes. Okay, so this is okay. So that's the story of he was a figure larger than life, to put it mildly. Now, when it comes to neuroscience, no talk is complete without mention of Ramonica Cajal and the controversy with Emilio Golgi, because before in the early days of brain histology, we used with the standard red and blue stains was difficult to fathom, and Golgi's claim to the fame was discovering what he called the silver nitrate stain. Okay, and with which the nerve fibers were stained dark black, and people were able to identify. And the earlier thoughts were that the brain was big continuation of network with fibers running like telegraph cables, because that was the technology that was up front in scientists' brain. And but Ramonica Cajal was challenged the doctrine. He was a microscopist par excellence, and was an artist, and had managed to paint the meticulous work of neural networks. And Cajal's drawings are still considered exquisite, and he showed that the brain had individual cells, the so-called pyramidal cell, which had nucleus and cell membrane, and the dendrites, an axon extended from the network. So he showed the different types of cells that were existent, and Golgi stood his, refused to believe the observation that Dr. Cajal had proposed. And they still, even when they shared the Nobel Prize, he continued to argue for the so-called reticular formation of neuronal network. Okay, but eventually, now at the same time, there were other people who are working on the Russian scientists. By this time, Russia was advancing on scientific front, and obviously a typical Russian name, Ilya Ilyevich Mechnikov, had proposed the phagocytosis, had observed the action of white blood cells. At the same time, the controversy started between the so-called humoral immunity, which was advocated by Ulrich, who was the assistant of Behring. And this, the arguments went back and forth between Mechnikov, and turns out that both of them were correct in their own manner, that the body uses both mechanisms for immune generation, the cellular as well as humoral, which is the chemical. So Ulrich did not buy the phagocytosis, and Mechnikov was hard to be convinced about the other part. So the scientific controversies continued for several years. Okay, now Ulrich went on to do more work on antimicrobial therapy, because syphilis was very virulent for whatever reasons, in the early part of in the 19th century and 20th century, that it was there was almost a whole chair of syphilology that was in vogue, because it affected the entire body, and for several decades, starting from the early infection called chancre, it affected not only heart but brain, and after several decades affected the brain and caused the insane, or the general paralysis of the insane, which was a really devastating illness. And with the rise of chemical industry in Germany, various dyes were being tested, and Dr. Ehrlich meticulously tested varieties of dyes, and finally was able to come up with a dye that was a derivative of arsenic, named salvarsan, and was successful in killing the tapinoma. Okay, and that was the start of antimicrobial therapy as we came to know it. So it's and this was later on, obviously, before the era of penicillin dawned. So this work was, carried out by his assistant Sahachiro Hata and Dr. Ehrlich, but Dr. Ehrlich was the one who, and this was important enough to be adapted into movie by Hollywood a few years later in 1930s. Okay, and again, to give an example of phagocytosis as to how the white blood cells, I think there's no sound in it, as to how the bacteria, which is interested in green, as the cell, which is basically protoplasm, engulfs the cell and eats it. So this is just the example of cellular immunity, and salvarsan, which is on this side. Okay, the early the German chemical giant Bayer company. Okay, all right. Moving on to this is, I'm sorry, my side's out a little out of sequence. This is the great Monica Cajal, and with this exquisite drawing, which are almost parallel to artwork. Okay, and this is again shows in more details the Golgi stain, the silver iron, and his shows a pyramidal network of the branching of trees of the cell nucleus at this in the middle. So it gives you an example. Okay, now sometimes surgical treatment, before the relationship of iodine and hormones became known, in areas where iodine was deficient in the mountainous area, people used to get a big swelling in the neck called goiter, and this was, and had a varicose effect of on their general metabolism, and had systemic effects. And so when the thyroid surgery was done, Dr. Kocher created, the people became lethargic, started gaining weight, their whole appearance changed, they were cold, and they eventually became retarded. Okay, and that's the derogatory term that was in vogue called cretinism. Okay, and so he was horrified with what he had created by doing a total thyroid surgery, and then decided to leave a small amount of thyroid as a residual, and then it showed that the small amount of thyroid, obviously they were not aware of what thyroid hormone's action was or what it was. It wasn't isolated, and so this was, and then subsequently it was treated by animal thyroid gland implant, and so they were able to avoid the disastrous sequelae of thyroid surgery. And obviously that was recognized as a Nobel Prize worthy work. At the same time, all of you are aware from high school of the Pavlov works about the so-called conditioned reflex of the dog and gong, and he used the metronome for dog to salivate. And then he carried out experimented to monkeys and even in orphan children. And no, obviously there was no actually human experimentation ethics, so Dr. Pavlov was not about using, putting children and enticing them with cookies and trying to see whether they can be trained with conditioned reflex with the gong or pressing up a rubber ball in the hand, so to speak, and at the same time, he was not about, commercializing his own discoveries of selling digestive juices, which he had collected as a therapeutic cure. So some of the stuff that he had done was really bordered on highly unethical behavior on children. So, but he became the founder of At the same time, a person who had worked with a tailor's apprentice, Alexei Corral, later on went to become surgeon and had pioneered the microscopic surgical techniques for suturing arteries and veins and anastomosis. And he was recognized for his work as a founder of vascular surgery. And later on the side, he was doing research on origin of life, and had worked on chicken cells, and he was able to keep them alive for a long time, and so-called perpetual chicken. But obviously that work was a little bit over the top, and maybe they were putting in new cells, so that was not the actual founding of stem cells as we know it, although it was suspected to be so. And on the side, he was a product of his time and was a eugenics supporter and a friend of the well-known aviator Charles Lindbergh. So at the same time, in the early days of 20th century, Freud, who started out as a neurologist, was instrumental in forming his own theory about the theories of mind and dream analysis, and later on moved to Britain. And this is the famous psychoanalytical couch. He was nominated for Nobel Prize, but never actually got the nod. Okay, and he still has a museum of his consulting rooms in Hampstead, London. Okay, now tropical medicine was big, as I mentioned earlier, not just Ronald Ross, but trypanosomiasis, which is the sleeping sickness that was prevalent in Africa and quite dangerous, with severe organ failures that can occur with liver and brain. And so that was the parasite needed to be isolated. Initially, even beriberi that occurred in Southeast Asia, that was suspected to be mosquito related, but later on that ideology was related, the correct cause was attributed to, that was related to the rice, the polished rice, rather than, and so the essential nutrients were missing, and there was the rise of vitamins as we came to know it. Okay. And now the malaria is, again, it's a very prevalent disease, and the malaria cycle is worth spending a minute or two. That the malaria bites a person, and it goes to the liver cells, and that goes through various forms called schizont and trophocyte, and it forms in the blood. And finally, when the schizont matures into, malaria has both male and female, and it's the female that is, and so it comes from after the red blood cell. It becomes, the so-called sporozoite cycle, and it's the female that bites and continues the cycle of reproduction. So it's a very complex, cycle that forms. Okay, and I think we are coming to the end of this thing, and I have recorded for almost, like 1.5 hours. So we'll pause at this stage. We'll stop the recording as it's a day through, and we'll continue next time. And okay, well, I will stop the recording and the meetings. Okay.